Hi, everyone. Thank you for tuning in. I'm Adam Lewis, our head of LQM research here at SandboxAQ on our AI simulation team. I'm here with Zane Beckwith, one of our scientific leads, and Paul Abruzzo from NVIDIA. I'll just pass to Zane to quickly introduce himself.
Hi, everybody. I'm Zane Beckwith. I'm the technical lead for drug discovery R&D at SandboxAQ, and one of the lead authors of the SAIR dataset project. And I'll pass to Paul Abruzzo, one of our collaborators from the NVIDIA team on this project.
Hi, folks. I'm Paul Abruzzo, product and engineering lead for NVIDIA's AI accelerator team.
Yeah, thank you. So we're here to talk about the release of our SAIR dataset, the structurally augmented IC50 repository. We're very excited about this. We believe in its ability to really revamp some of our ability to predict drug potencies and structures at an unprecedented scale. And we're going to go into that over the course of this short webinar. First, I just want to talk a little bit about SandboxAQ. We're a science-first technology company. We come from Alphabet from 2022. And what we really focus on is tackling hard scientific problems at the core of B2B impact. That is, scientific problems that are valuable commercially and having a big impact on a humanitarian scale as well. For AQ Biosim, which is a subset of SandboxAQ concerned with developing better therapeutics, our mission is to accelerate drug discovery by unifying machine learning, computational chemistry, and biological reasoning, moving the early, high-risk, most costly steps, actually all the steps, in silico. We're backed right now by $950 million from some top-tier investors, and we have a strategy of partnering with pharma, biotech, and academia to deliver maximum impact. We're engaged right now with over 18 pipelines, spanning really across all therapeutic areas, modalities, and client types. We can see some of those here. We have some work in drug discovery, some in clinical development, in a variety of different indications, neurodegenerative disease, oncology, autoimmune, and really kind of every client type profile, big pharma, early stage, academia, nonprofit, you name it. What we're really aiming to do is deliver broad impact, deep impact at scale, as we like to call it, across the pharmaceutical pipeline. So here at SandboxAQ, we're really seeing ourselves as being at the cusp of an opportunity to revamp drug discovery by injecting AI and computational methods into really every part of the phase. And underlying that is, of course, the need for advanced AI compute and for expertise both in driving that compute, using it, scaling it up, and then, of course, in applying it towards the scientific problems in drug discovery. So to deliver on that, we've been partnering with NVIDIA, and it's been a very close relationship. They've been really very instrumental in helping us take some of the scientific results and impact that we want to make and scaling it up, making it possible and interacting with us really kind of at every step of the way to help us realize and deliver on that vision. And SAIR is an example of that. But we also have several other collaborations with NVIDIA that we'd like to display. You can see them on the slide here. What we just want to say is it's been a very close and beneficial partnership that we see delivering impact both now and in the future. So, to get started, our vision here at SandboxAQ is really to enable AI to deliver on enhancing the research productivity, that is the number of new drugs delivered per unit time, if you like, with unprecedented efficacy and at an unprecedented rate. We see kind of like three pillars to AI that we think can add value here. On the far left is agentic AI, which we believe in its ability to formulate new experiments, really reason kind of in a forward-looking way about what tools to use, what algorithms to apply. On the far right, we have our causal models. These are models that we use to take existing data, whether it comes from our own pipelines or experimental ones, and try to unearth cause and effect relationships in them. And on the middle is the models, and this is more what we're talking about here, that we call LQMs. And this is a term that we use to describe scientific models that are not LLMs, are not trained on text, but rather are trained on scientific data to give real quantitative precision for predictions in scientific and quantitative tasks. So our vision is that to use all of these in concert, processing data with the causal models, reasoning about what to do with the agentic models, and predicting the results of experiments that might be too expensive, too costly, too inconvenient, or simply too slow to do with the LQM models. And that's our vision here. So just to give a brief overview of how we think about these models interacting with the pharmaceutical pipeline, I just want to very, very quickly go over a high-level version of the small molecule drug discovery process. This starts on what we've depicted on the far left as target identification. So somewhere out there, people have identified a humanitarian need, a disease that either hasn't been treated or isn't being treated adequately, and a protein usually in the body, not necessarily a protein, but for the small molecule process typically, a protein that if bound to with a drug would modify the disease. We then move on to the drug design stage, which is concerned with having been given that protein, finding a molecule to strongly bind it in a way that has various concerns addressed, such as safety, but a big bottleneck to this is finding the drug binding and potency in the first place, and this is what SAIR is going to address. And then finally, once this molecule is identified, we move into drug development, which is concerned with convincing regulators that the drug works and is safe. A SAIR is really oriented towards the drug design part of the pipeline, and the bottleneck here, or a major bottleneck, is in quickly finding new strongly binding molecules to a given protein. You're given the protein, you need to find ideally a variety of different molecules that are really good binders to it, because those identify regions of what's called chemical space that will allow you to develop new drugs from. So now I'd like to go on to introduce SAIR more directly. So SAIR stands for Structurally Augmented IC50 Repository. It's a data set that we've developed to really enable our ability to find new high potency binders to proteins very quickly and accurately. And what it specifically is a data set of 5,244,285 computationally folded 3D drug protein molecules co-structures tagged with experimental potency data. Let me just repeat that in simpler terms. So it's a list of, we have a list of different proteins. These proteins are given as sequences, that is the chemical structure of the protein, paired with small molecules, and paired with a measured potency for each. So we take those, there's about 1 million of them. We then apply what's called a cofolding model. In this case, we use one called Boltz-1X to go through those different proteins and develop a 3D structure, such as those you see on the top right, that includes both the protein and the small molecule. And we actually create 5 such structures for each pair we have. Those structures differ a little bit because the model that we use doesn't have perfect knowledge of what the true structure would be. And the regions of the protein that differ from each other most are the ones where the model has the less certainty. So what this data set does is allow us to compare these 5 different results against other means of predicting potency computationally, and therefore to select from them which ones are the best for further enhancing these sorts of potency predictions. So in short, it's a data set that allows you, using only structure affinity data, to go on to enhance models for producing these 3D structures in a way that doesn't rely on having direct measurements of those structures in the first place, which are slow and expensive. So what part of the drug discovery process does this speak to? So we call this the potency optimization loop. And in very simple terms, what one would do in computational drug discovery traditionally is first you find a 3D structure for a protein. So you're given, you know what protein you're going after. It's been identified in the target discovery phase. Now you want to know what its 3D shape is, as depicted on the left here. And that would traditionally be done by very slow and expensive experimental methods. Now, if you're given that structure, next you're going to go on to find the pose of a drug molecule. So we'll be given a big list of drug molecules to scan over, and we want to find, as depicted again, where the arrow that says drug molecule is pointing up, where in space that molecule is oriented and basically stuck to the protein. And finally, once we have that information, we want to go on to find the affinity of the molecule in that pose, that is how strongly it's sticking to the protein. And that last number, the strength of the sticking, is correlated with the actual potency of the drug and ultimately its efficacy. So for some time, the AI drug discovery community has been really focused on, among other things, but one area of focus has been on taking that potency optimization loop and bringing it in silico, making it a computational process. This has started off with what are called folding models, and these models started by really taking step 1 and trying to make it a computational thing. So AlphaFold is a famous example of that. What these models did is continue to do, is they're given a sequence and they give you a structure, but they don't give you the structure of the molecule. Absent that, these models, although very interesting and useful for many purposes, struggle to interface directly with the potency optimization loop because the accuracy of the subsequent pose and affinity tends to compound in error and turns out not to be as accurate as you might like. The next stage, which came out around with AlphaFold 3 especially, and the Boltz 1X model we use, are called cofolding models. These models fold the protein and the molecule together as one, and therefore come up with both a pose prediction and a structure prediction at the same time. And this further improves the accuracy of the final prediction, although still to a point where there can be errors, especially when we're dealing with proteins that are quite different from the ones in the original data set. And finally, there's a new wave, just not really a wave, just one now, Boltz 2, which integrates all of these steps into one and produces a joint affinity prediction at the same time as it gives you a pose and a structure. The jury's still out here, but although these models have very impressive results, their ability to generalize is still suffering. That is, we've kind of already trained everything that's publicly available in creating these models, and there isn't really much more direct structural data to use, which means that when you try to run these models on something that's very different from what they've seen, they often struggle to produce accurate results. So where to go forward there? How do we keep going? How do we go on to make models that can generalize? And one means to this end is SAIR. So SAIR allows you to leverage affinity data for structural predictions. So instead of structural data, which is relatively very expensive to make, SAIR is a way of taking affinity data, that is just having the sequence of a protein and a molecule and knowing how potent it is, and filling in, if you like, the intermediate step, the structure, in order to allow that data like that, which is very relatively cheap to make and abundant, to be directly leveraged to improve cofolding models. The way we did this is we took these sorts of data sets that have sequence and protein matches, like Chembl and BindingDB. We went through them, and then we created the structures with AI, that is with cofolding models, on about 1 million examples where we had the sequence, the molecule, and a labeled potency. We ran those through the Boltz 1X cofolding model on NVIDIA DGX Compute, which allowed us to do this at very favorable scale, speed, and utilization. And we output 5 different structures for each molecule. With each of these 5 structures, which differ because of the numerical deviation between different results, we can then compare with other potency predictions or measurements to select from the best of each. And in this way, we can refine the models using only potency data. We generated this data set on NVIDIA's DGX Cloud AI platform, and that enabled an amount of data generation that we had originally scoped for a whole quarter of work to proceed over about 3 weeks, give or take. It was really quite a dramatic improvement. And that's a lot of thanks to NVIDIA's platform and team getting us up to the utilization we needed, and their platform enabling integrated training of models over SAIR as well. And I will now pass to Paul Abruzzo here to speak a little bit more about the NVIDIA platform.
Thanks, Adam. Now let's talk a bit about how NVIDIA's DGX Cloud team and SandboxAQ partnered to achieve scale-out results at record pace. DGX Cloud provides managed short-term capacity, backed by NVIDIA's expertise. It's built on top of Tier 1 cloud service providers and NVIDIA's cloud partners, managed services, combining the best of NVIDIA infrastructure and software technologies. We work with you to make sure, well before you hit the ground running with scaled capacity, that a full set of outcomes are defined, your data sets are ready, the environment is configured and hooked into your internal systems, and that the infrastructure is secured with CSP, NVIDIA, and your organization's best practice, whether it be training or tuning, so that you can achieve time to first train at record pace. The goal is to establish the necessary patterns and playbooks covering DevOps, FinOps, MLOps, and engineering that you can take away from your engagement with AI Accelerator and into your own managed infrastructure, whoever your hyperscaler of choice might be. Now, let's talk a bit about what the AI Accelerator team was able to do for SandboxAQ. Success for Sandbox meant first securing capacity, appropriately sized for the needs of the well-defined engagement phases. This meant looking to DGX Cloud in Google Cloud to work closely with Sandbox's existing infrastructure. Over the course of less than 30 days, we worked with Sandbox to scale from 280 to 768 H100 GPUs as workload demand increased. Throughout the engagement, the NVIDIA AI Accelerator team closely monitored all aspects of infrastructure performance, looking to node, operator, scheduler, and GPU metrics to identify bottlenecks, implementing and optimizing with managed services like GCP Parallel Store, and job framework optimization to achieve the highest effective utilization. We also work directly with SandboxAQ engineering and research teams to optimize GPU workloads, identifying bottlenecks, for example, CPU-bound calculations, and working with domain experts at NVIDIA to help increase effective utilization and time to result. Over the 130,000 H100 GPU hours, we were able to achieve greater than 95% effective GPU utilization throughout the engagement. It's really about enabling AI Accelerator customers to do what they do best. We wanted Sandbox to focus on their core competencies while we handled the complex GPU infrastructure, scheduler orchestration, configuration, and support. Now, back to you, Adam.
Thanks, Paul. So now I'm going to just give a little bit of an overview of some of the early scientific benchmarking that we did on the SAIR dataset. I'd really encourage all of you to take a look at the manuscript that we currently have on the bioRxiv. It's displayed here, and we'll provide a link. But I'm just going to give like a little bit of a teaser, if you like, of some of the results in there. So an early example application is that we use just to kind of show that the SAIR dataset can meaningfully interface with our existing models for affinity prediction. We use it to benchmark those. So SAIR contains entries from different kinds of assays, like cellular assays and other kinds. And what we did is we took the entries in the data from different types of assay, and we compared them against other ways of predicting affinity computationally. So on the bottom of this bar chart, each tick represents a different kind of computational potency prediction tool. If those models are accurate, then they should produce, ideally, highly correlated results between different types of assay, because each type of assay would have, although they differ sometimes in some ways, at the end of the day, our measurements, including the core affinity prediction that these computational methods are producing. And what we found, just to kind of cut to the chase, the best correlations we found were from the two deep learn models displayed here, OnionNet and AvePill. These models are themselves machine learning method, which is kind of promising for the basic idea of AI drug discovery, finding that the deep learned, more data-driven methods are showing better correlation and better results. So we were impressed by that. What's next with SAIR? So the biggest thing that we'd like to do is improve the coverage. Really, what we see this as is a path forward to it really massively enhancing the amount of data available to the community. So we'd like to start repeating the process for different kinds of molecules, different modalities, which maybe aren't necessarily small molecules, such as antibodies, and finding different data types apart from simple potency measurements to extract similar signals as a way of, again, being able to leverage all the diverse and large amounts of data available to the biopharma community for these predictions, given that we've exhausted the direct measurements. Finally, what we're really excited about as a company is connecting results from this with whole cell signaling, because we see a lot of promise right now in modeling biology at a larger scale than simply molecular, and we think that's where a lot of the impact is going to come from in the future. So to use the data set and learn more, SAIR is, completely free to access. You can see the manuscript, blog post, and data set at the web link displayed at the bottom left. For non-commercial users, there's no restrictions at all for use. For commercial users, we don't intend to make any restrictions. All you got to do is submit a short form to us, and we'll respond to you with a license for commercial use. We would love if you could just tell us a little bit about your use case, because we're interested in seeing what kind of impact the community is having with this data set. And now I will pass to Zane Beckwith, who's going to show a bit about how practically to access SAIR and what that really looks like.
Thanks, Adam. So I just wanted to show what the data set looks like, how you can access it, how you can bring it down locally to be able to work with it yourselves. So just as a quick overview, this is available on Google Cloud in a completely publicly available account and bucket. As it stands, it's about 760 gigabytes in size. That's because we've applied quite a bit of compression to all of the structure files and things like that. Again, in total, once it's uncompressed, it's about 2.5 terabytes of data. But the 760 gigabyte size makes it much easier to download and manage. The analysis files, the analysis that we've done, the predicted affinities, the original experimentally determined IC50 values, various metadata such as the model confidences, PLDTs, things like that, as well as just a lot of metadata about the source of some of the underlying experimental data. That's all available in a Parquet file that's in the bucket under SAIR.parquet to allow any kind of data science, data analysis that you might want to do. Those raw confidences and PLDTs are also available in their own gzipped tarballs to access. We also have instructions on how to download the data, the structure of the data, columns of that Parquet file, things like that in these README files. And the meat of the data set is those predicted structure files. Those are in the structures compressed folder. Again, this comprises about 100 or exactly 105 gzipped tarball files that make up, once uncompressed, the full over 5 million CIF structure files. So you can see that here. Let me show you what it looks like in the bucket itself. So you can see all of that here in the bucket itself. It's publicly available. Again, you can see everything that's available. And again, if you click on the compressed structure directory, you can see there are quite a few of the 105 files. These are organized by the entries that show what the protein ligand complexes are that were folded, and that can be tracked back to the metadata in the Parquet file as well. But now something that's pretty cool about this, because this is being hosted in Google Cloud, you can download this Parquet file directly and load it via Pandas or however you might want to look at it. But we also have it uploaded as a BigQuery dataset in the same publicly available account. So you can see here, as an example, what the columns are that are available there. You can use all the niceties of BigQuery for, complex SQL-based, highly performant queries against this data set to downsample, subselect, do whatever you might want to do. But the BigQuery interface is quite nice for handling something at this scale. Again, this is 5 million lines in the data set, so 5 million rows in the data set. So it's quite nice to use BigQuery for accessing it that way. So we can see here how to pull the data set down to a local machine you might have. As an example here, I've just got a VM that I spun up to show this. I'm going to execute the gcloud CLI to pull down the full data set. The rsync command is really useful for that, to handle on-the-fly compression and things like that. So we just say, all right, pull down that existing public bucket, and you'll see here that it will start reporting metrics on bandwidth usage and how much data has been downloaded. Now, again, in total, this is about 760 gigabytes, so this can take a few hours. We won't sit here and watch the whole thing tick through. But you can see it starts to download all of the various tarballs and is giving progress metrics on how far you have to go. So let's look then, once we have the data set downloaded here. So this is just another copy that I have already downloaded to this machine. Again, we see it's exactly what we just saw in the Google Cloud console. We've got the license and the instruction readmes. We've got that Parquet file, the confidences, the PLDTs, the manuscript, and again, that structures compressed directory, which has all of the tarballs for the raw CIF structure files. So if we want to decompress all of those tarballs holding the structure files, this is an example of how you might do that, just piping it all through the standard Linux tar command to decompress all of those 105 structure tarballs. So we see here, again, it's reporting that it's pulling out all of the CIF files. Again, because this is over 5 million files, this decompression and untarring takes a little while. So we won't sit here and watch it all tick by. But once that completes, then you'll have a structures directory that has all of the raw CIF files in it. And you can do great open science with it at that point. So excited for everybody to use this. I hope happy trails with downloading it and using it. I'll pass it back now to Adam. Thanks, everybody.
Thanks, Zane. And just to close off, we're so excited about what the community is going to do with this data set. We want everyone to have it in their hands, on their laptops, posted to their fridge. It's, we think, really something quite game-changing. We are very excited to see what you're going to do. So please access the data set. You can find it at the bottom left here. If you're a non-commercial user, you can just download it, use it right away. If you're a commercial user, no problem. Just fill in a short form, tell us what you're going to do with it, and we'll send you a license right over. We really want to see what the community is going to do here. Find us, email us, contact us by LinkedIn, however you want. We are really excited about this, and I just want to close off with that, and thank you so much.